All right, good afternoon, and thank you all for joining us this close to the end of the conference. My name is Becky Kapp, and I am with BioBridge Global. Thanks to my friend and now colleague, Sean Thompson, who brought me in over the summer and started working with him and with the BioBridge team. I come from a pharmaceutical background, and so getting into the world of blood centers and cellular therapy took me all the way back to my first position in the medical space, where I worked as a clinical data manager for Dana-Farber Cancer Institute on one of the big trials in breast cancer, looking at autologous hematopoietic stem cell transplant versus high-dose chemotherapy back in the early 90s, trying to answer that question. So coming back and looking at what is going on at BioBridge and trying to understand that so that I can help move this mission forward. My anchor was in that cell therapy space. One of the things that I have been particularly excited about is looking at how this field has developed and the new places it can go. I went to consulting after my time at Dana-Farber, and so the transition from maximizing revenue to maximizing good while supporting revenue is a new framework for me, and it's sparked a great deal of creativity in the discussions that I've had with the rest of the leadership team at BioBridge and with my contacts in the field. And I think that is really what we wanted to focus on in this panel discussion today. We're going from being service providers of standard available services to feeding an entirely different industry, an entirely different line of clinical and scientific exploration, and that requires some creative thinking. So with that, I wanted to introduce our first speaker. Linda Myers has an incredible wealth of experience in blood banking and cell therapy arena. She started at the bench and worked her way into management, taking on roles in quality assurance. She has had a significant leadership role in Qualtex that eventually evolved into the leadership role as the COO of that division. In 2013, she took over as the CEO of what at the time was South Texas Blood and Tissue Holding Company and evolved that into BioBridge Global. So the creative leadership that she has exhibited in taking BioBridge to where it is and to new directions has been a real inspiration, and I'd like for her to talk a little bit about that.
Thank you, Becky. Really appreciate you being here today. I wanted to share with you a little bit about why BioBridge Global has supported the World Stem Cell Summit. Okay, I'm getting a signal here that this is not working. This is our third year to support the Stem Cell Summit because we recognize that the blood banking industry has a lot to offer in the area of regenerative medicine. Blood transfusion has been touted as one of the first successful tissue transplants, and we have been perfecting this for decades. Are we working? Okay. So I just wanted to take just a few minutes just to give you a very brief highlight of why blood centers and tissue centers are uniquely placed to be a participant in the regenerative medicine field. So BioBridge Global has been in the blood banking industry for over 41 years. And we have several affiliates in our organization. We're not-for-profit. We have a blood bank, a tissue bank, a cord blood bank, a large testing lab, and a foundation as well. But collectively, our mission is to save and enhance lives through the healing power of human cells. And we have recently just broadened that mission scope for our organization. So we feel like that historically we've been involved in cell therapies, that we really want to take our mission to a new level And that new level includes enabling research and the field of regenerative medicine, cell therapy and regenerative medicine. So the question at hand is, why do advanced blood and tissue centers feel like that they can contribute to this industry and to commercialize treatments in the field of regenerative medicine? We also know that in our field of blood banking, things have changed, and the focus is moving away from our historical duties. And we have been in a field which typically has had low innovation and has been very reactionary to supplying blood and blood products. So we're very committed to our mission, and we want to drive this forward. We have access to a lot of biomaterials that can enable and support research. And we also have decades of experience in fine-tuning core competencies that are needed in the research industry. And it is a clear fit to regenerative medicine. So how do the advanced blood centers support that? I have some very esteemed colleagues here today who are going to share with you some examples of the types of things that we can contribute to. We have a wide array of human biologic materials that are available. We have facilities that are GMP compliant, and at BioBridge we maintain ISO certification, EU certifications, and those certifications with tissue and blood industries. We're experts at dealing with the FDA. We've been doing it for decades, so we have a high regulatory and compliance expertise. We're manufacturers, and we're continuing to expand that manufacturing capability. And we're partnering in the industry to help move forward the scientific, clinical, and commercialization of treatments for patients. Our ownership spans a whole wide spectrum of offerings, from the very basic biomaterials to fully integrated solutions. And we do that through tissue supply, quality testing, assay development, new ways of engaging donors. You're going to hear something about that today. And multi-site research and collaborations. Those multi-site collaborations ensure consistency of standard operating procedures and also helps to address the stages of development of procedures or research projects. I feel very fortunate being in San Antonio because we have a very strong regional presence for collaboration. We have the military, medical, and research facilities concentrated in San Antonio. We have 2 large research institutions that we work with, along with a medical school and a university. And to wrap that all around, the city of San Antonio also supports a nonprofit organization called Biomed SA, which 100% of their focus is to take all of these pieces within our community and facilitate collaboration. And it's been a hugely valuable thing for BioBridge Global. And so I want to call up my colleague, Dr. Scott Jones, who's going to tell you about a couple of the collaborative efforts that we've been involved in our community.
All right, thank you. Good to see people here. I know it's late in the meeting process, so it's good to at least see some people out there. What I'm going to do is just sort of expand a little bit on what Linda already laid a great foundation on, and that is tell you a little bit about specifically what kind of collaborations we're doing, and specifically novel collaborations related to stem cell research or supporting stem cell research and development of stem cell products, specifically at BioBridge Global. So Linda already showed this slide, but what I wanted to sort of reiterate is this is really the mission of all of the business units here at BioBridge Global. And really what we're really trying to do is enable stem cell research and develop, and also enable the development of stem cell products. And so really how we do that are sort of those core competencies that Linda already mentioned. And The two that I think are kind of the theme of what I'm going to talk about today are obviously one is the regulatory core competency that we do have. We are very CGMP compliant. Like Linda said, we work with the FDA. I know there's a few blood banks here, but I don't know if you guys realize, but we get audited anywhere between 25 to 30 times a year. So you can do the math, how much that is per month. So we're very used to the regulatory environment and able to be prepared and support that. And then the other sort of core competency that is near and dear to my heart, and that is assay development. Being able to develop specific assays. That's kind of always been sort of a trademark difference of our blood center and of BioBridge Global. So specifically in the blood world, we've already developed 3 different assays, really from the research part all the way through development, and also working with the FDA to get them out there. And we currently use these, at least 2 of the 3 assays. So we really wanted to sort of take that knowledge and utilize it with our collaborations. So to go ahead and start, one of them, and I'm going to go through 6 different collaborations, and these are obviously at different points. Some of them are just beginning, and some of them are projects that we've been working on for quite some time. One of the things is to sort of talk about cell characterization, and so we have just now embarked on a collaboration with an international multi-center group. So kind of like what Linda mentioned, that a lot of these are involving centers throughout the nation or throughout the world. And specifically with this one is we're bringing in expertise of a German researcher who has very big knowledge or large knowledge base for mesenchymal stem cells, and has experience not only in utilizing them but also in isolating and being able to expand them. And then we're combining that with a research group that's really kind of a research clinical group, so it has the clinical expertise, and then obviously the expertise of BioBridge Global to include the GMP and also the assay development expertise. And the idea is really sort of to work together to kind of take, there are some published international standards and there are some guidelines, and I know from a discussion yesterday that there's an NIST standard coming out soon, or at least a guideline. But really sort of working on those and seeing what we can do as a group to come up with sort of a general best practices for this group. So we can make sure that when these products are being developed and being worked on, that we're basically hitting all of the regulatory and all of the components that FDA is going to need. So the bottom line is we're able to get products out quicker and actually get a product that we could get to market within a reasonable amount of time. The other one under cell characterization is to talk about 2 collaborations with biotech companies. One of them is sort of, or both of them are related to really looking at characterization and potency assays. So once again, I mentioned that one of our core competencies is that we do assay development. So what we're interested in is if we could develop an assay to basically characterize specifically, mesenchymal stem cells or other cells, that way when people are doing or designing their testing protocols, whether that is preclinical or clinical, they're basically able to go ahead and use cells and be able to reproduce it. So really what we do on a day-to-day basis within the blood banking world. And the other concept then is you could also use an assay like that to show potency. So if you could take that concept or characteristic of it and basically create some kind of fingerprint, then you could utilize that. Also, if you could show that it's related to the actual biologic activity, then you could utilize it as a potential potency assay. And I know I've heard numerous conversations, and having run a round table, everybody was very worried about what are we going to do about potency, how are we going to show that. But the concept then is if you could do this early on, you could also address it early on, so that way you're not going to have a great surprise at the end of the road when you're trying to actually get a product out through the FDA. So 2 of the things that we've been working on for a couple of years are, one of them is looking at microRNA. So the concept is if you could look at differential expression of microRNA within a cell, you could probably create a fingerprint, so really kind of like a specific pattern that way you could tell and utilize sort of your criteria to say that you've got a similar cell group that you're using throughout your process. And like I said, you could possibly use it for potency if you can show, hey, as long as it's representing or showing this pattern, we see that we have a potent product. So we have been working on that for a while. You can sort of see, I just want to show you at least just a general idea of what I'm talking about. And so there's a little graph here. And what you're looking at are really 4 different colors, but really same starting cells, but these cells have been kind of grown in different ways. And as you can see this is just a visual representation of the differences in microRNA expression. And you can tell that you actually have different subgroups based on that. So that's sort of the concept that we were talking about. The other thing is, instead of just looking at microRNA, because obviously, if you look at these things, they're very complex. And so you're probably going to have multiple ways to look at it and maybe multiple assays to show it. So another thing that we're doing with a local biotech company in San Antonio is they actually have a quick and easy assay where they can sort of analyze the membrane potential, specifically porn proteins and the movement of ions. And I know it sounds a little crazy, like why are you looking at that? What does that really have to do with the cells? But being sort of a bacterial physiologist in my beginning, the concept is that if you can understand that and if you can look at that potential, you could also possibly use that as a fingerprint, as a specific pattern that you could look for, once again, for characterization and also for the possibility of potency. So that's another avenue that we're just now beginning and looking at. So to sort of change gears, we're also looking at collaborations related to expansion. So specifically in this case, looking at trying to create a new source of stem cells for research. So another thing that our group has done in the last year is basically do kind of voice of the customer. Got everybody together from the community, from researchers, military, large academic institutes, local biotech, and really said, hey, what do you guys need? What are those hurdles? What are those things that we can do to help you? And one of the things that came out was, hey, what we could really use is a good source of stem cells. Well, it turns out we have a cord blood bank. We have samples that we're not able to bank. So the concept is take those, isolate the MSCs from cord blood, and then basically expand them using our good friends at STEM Biosys using their technology, and then also further expanding it so we can get enough cells in order to help or be enough potency for the research project using a bioreactor that we have at BioBridge Global. So that's another collaboration that we're doing. The other concept too is, we can work in cellular therapy, but what we can also do is, once again, use our core competency. So in this case, it's a company, a biotech company that has an expansion process. They have done it within their laboratory, but what they really need help with is, hey, how are we going to basically scale this up? How are we going to do this in a real production environment? So what we're able to do is tell them, hey, come on in, bring your stuff in, and we will basically collaborate with you, and we will validate your system, help you with SOPs, help you basically get that system up and going. So really providing that bridge that's able to help them through that process. And then really the last one real quick is working on this sort of cool clinical center of excellence. So what this is really a large group that we're putting together. One of them is looking at or getting scientific expertise from a local academic institute. And then also combining that with, we're fortunate in San Antonio to also have a research institute that has a large non-human primate model. And then bringing in the core competent clinical center from BioBridge. So really it's not just the concept that we could do the testing and all that, but what we can also do is do the collection of these cells. We can basically then do the processing, the testing, all of that related to it, and even administer that product and basically try out this novel platform. So really being able to develop this whole new treatment program, which basically could be used for varieties of diseases, but being able to then develop that with the help from BioBridge. So I just wanted to say thank you and letting me come up here and speak, and I hope you have a better idea now of really the breadth and the variety of collaborations that can be done related to stem cell research and also development of stem cell products that we're doing specifically at BioBridge Global. So thank you.
Thank you, Scott. So I didn't introduce you properly. I'll tell the crowd a little bit about you. Dr. Jones has been with us since 1999, and his background in microbiology and infectious disease has been incredibly valuable. But what I've come to value about him in the last few months of interacting with him is what an incredible collaborator he is, very thoughtful, creative, and very easy to work with. So I think that can't help but help us out in that regard. Next, I'd like to introduce Dr. Rob Tressler. He's the Vice President of Cell Biology and Therapy at San Diego Blood Bank. He has more than 20 years of industry experience in preclinical research and development. He brings with that expertise in preclinical pharmacology. He's held positions of senior leadership in a number of pharmaceutical companies, especially in oncology, and he has an extensive publication list and is an inventor on a number of patents. So thank you, Dr. Tressler.
Thank you. And thanks to the folks here who have the tenacity on the last day, on the last afternoon to stick it out. And all that stuff she said about me is not a big deal. When you get old, you start to accumulate things, you know. So that's the way that works.
Actually,
how do I make this go?
Yep, sorry, I thought I'd hit that.
I'm so old that I don't know what computer. A computer to me is still a TV with a typewriter in front of it.
There we go. Yeah. It's not a mistake. Sorry.
I'm going to give the two speakers before have just done a beautiful job of laying out what as a subset we as a blood bank can do at San Diego Blood Bank. So I'll go through some of my slides pretty quickly, and I'll speak to some specific examples and experiences that we've had and are currently going through right now. Stem cell, we're at this meeting. Stem cell therapeutics are an exploding field, and in fact, I won't belabor this point, but there are some various uses that we have direct experience with that I'll speak to a little bit further into the talk. But obviously, and regenerative medicine, transplantation, oncology, and immunology are all areas of stem cell therapeutic potential. Some approvals have occurred now, thank goodness, to validate the concept beyond just regular transplantation, which there's nothing wrong with that. It saves thousands of people's lives. Some of the things we've worked with specifically are fetal layers, cytokine production, and cell product progenitors, as well as growth factor supplements, which I didn't list, and data. Yesterday I was corrected. We're now over almost to 600 phase 1, 2, and 3 clinical trials just with mesenchymal stem cells worldwide. So it's an exploding field. If you look at where we were back in 2000 versus where we are now and are going to be in another 2 to 3 years, it's a remarkable growth of this field. We're still struggling with potency and identity, as was so elegantly mentioned in the previous talk. So that's one of the big hurdles we have to overcome. This is some of the potential products and services that the San Diego Blood Bank offers, and I think all blood banks can offer. Obviously, white blood cell fractions, whether it be PBMCs or mononuclear cells, and also red blood cells. RBCs are an interesting one for us. We actually have an early stage basic research collaboration with a startup biotech, and they're basically trying to characterize the aging of red blood cells when they're being stored for use. Right now they have about a 45-day half-life or shelf life, and then you have to get rid of them. Well, that's a shame, you know. I mean, blood's a very precious product. So we're in some very early stage work with a group that's actually applied SBIR grants. We've actually written letters of support, and we're moving forward on that front. Assay development, another startup company we're working with very closely now, is trying to develop a novel in vitro pyrogenicity assay that gets away from using animals for pyrogenicity studies. So it's an exciting front as well. And we collect a lot of platelets. And we throw away a lot of expired platelet units, or we used to. We don't anymore. We've now decided on our own volition to start generating a media supplement. We've been doing a lot of internal research on it to characterize and optimize it. We looked at what the community offers, and it's a very good product, But we did some work to optimize our platelet products, and we've made some novel findings with it and filed patents on it as well. And we're actually launching the product tomorrow. I'll be in San Diego at the American Society for Cell Biology meeting to share the information on the product with the world at large. But we've also offered it to the University of California at San Diego. They're evaluating it for its effects on phenotype, just to make sure that when you grow cells in it, the phenotype doesn't shift. Because one of the problems with expanding stem cells is you want to maintain the original phenotype of your product, or your API, as we like to call it. I head up the cord blood bank, so I have over 1,000 fully consented for research use cord blood units that are cryopreserved. And we generate more every week, because we collect anywhere from 20 to 40 cord blood units a week just in our local catchment area. And as you all know, 9 out of 10 of those are just because of total nucleated cell count not high enough to use, but they've been collected under CGMP. Viability has been assessed, T&C counts have been made. And so we've been offering to a variety of companies, Novartis, the UC system, and other companies, just cord bloods, processed cord blood units for research purposes. The caveat there is make sure you've got a good consent form put in place, that it's bulletproof. Make sure you've been transparent with the people. And that's one of the advantages of blood banks as a research collaborator with biopharma as well as with academics is, as was mentioned earlier, the FDA is all over us. We don't get quite that many audits, but we get about 12 to 14 a year. So we're on a first-name basis with the agency as well. And one of the things we've had to be very sensitive to is consent, and IRB clearances for research use. And we're doing stuff that's everything from very basic research out to phase 1, phase 2 clinical trial research in collaboration with a variety of companies. One of the more interesting research collaborations we set up was with the La Jolla Institute for Allergy and Immunology. And they weren't sure what they wanted to do with this initially. Well, they had some ideas. They said, well, we'd like to get some PBMCs from you and do some immunogenicity testing. One investigator there, and we said, sure, we'll do that with you. And he said, you guys, you have a great facility here, and you can generate a lot of products for us, and you can even fractionate it and give us different subsets. And we said, sure, what would you like to do? They said, well, we're not sure, so how about this? Can we compile a questionnaire amongst our researchers? And they have 10 or 15 different research groups within the Institute. And they went back to them and said, give us your questions that you'd like to have answered for potential collaborations and interactions with the San Diego Blood Bank because of the products they can supply, whether it be cord blood cells, white blood cells, red blood cells, buffy coats, plasma components, or platelet components. So they compiled a 45 question questionnaire, and we got clearance to submit it to all of our donors, and asked that those that wanted to participate that filled out the questionnaire. As we took it back to the investigators. They then collated all the answers that were relevant to each of the different functional investigative groups in the Institute. And now various investigators come and say, well, I'd like to get PBMCs from this cohort of people, de-identified, except for their physical or medical characteristics. I'd like to get samples from that cohort of people because of their allergy profile that they mention in their questionnaire and stuff like that. So we've put in place not only active reaching out on a one-to-one basis for specific research programs, but we've also put out, by doing this, if you will, a solicitation or an opportunity to a whole institute with different investigators doing different types of therapies to then come back to us and work with us. So that's been really good. We've also, and I can say this, it's now public, we've started a homegrown precision medicine initiative, if you will. It's a human genome project. Basically, it grew out of the notion that Barack Obama and others had posited about, learning the human genome will go a long way towards better precision medicine, both to decide who should get therapy and who should not, as well as giving a sense of what eventually is down the road for you, what you could be predisposed to as far as a disorder later in life. The problem with that in the past when people have tried to do it is the concern of confidentiality and HIPAA compliance. In other words, well, if I give you my DNA, it's going to go to the FBI, and if I ever do it, and I never would do anything wrong, but I don't want to be in a database where I can be identified. Well, as my CEO says, David says, is cord blood banks are like the Switzerland out there, because we've already been collecting human samples and analyzing and typing them for decades. and we're like a firewall between the man and the general population, if you will. So we are part of that. We have that trust amongst our demographic that we service. And in fact, we had a huge response from people saying, I'd love to be a participant in that. I'd love to get my whole genome sequenced, and I'd also like to have it available for query as long as I'm de-identified. It was remarkable. We thought, well, we'll get a few people. We got flooded with people. So in collaboration with Illumina, we're now building out that precision medicine initiative. It's at very early stages, and we've got a lot of technical pieces to put into place as far as storage, archiving, and de-identification, but it's moving forward. We also do internal work, mostly because I sit on a gold mine of STEM. I'm an oncologist / cell biologist, and I get, like I said, I get 20 to 30 fresh cord blood units a week that can't be listed because of low cell counts that have perfectly viable stem cells in them. So we've started some internal initiatives to identify those, and we've actually started discussions with externals about our work where we're looking at isolating and enriching for NK cells or possibly T cells, because everybody's a CAR T cell fanatic right now. So UC San Diego is actually reaching out to us to look into that type of collaboration. And we're also internally doing work on looking at reversing the senescent phenotype of mesenchymal stem cells. One of the concerns with MSC-based therapies that's autologous is if you're an old coot like me that's over the age of 60, you know, my MSCs are probably okay, but they're getting a little tired. If there were a way to revitalize them, perhaps that would make them more robust if I ever, God forbid, needed them for a therapeutic approach. I mean, right now we're doing clinical trials, and one of the criticisms is we take a person who has an autoimmune disorder that's 70 years old. Inherent in that is probably their mesenchymal stem cells, which are part of the immunoregulatory system, aren't working as well as they should. So we suck the MSCs out of that patient, we make a bunch more of them, and we put them back in and hope it's now going to work. Okay? Maybe, you know, if a certain subfraction of those cells are still youthful or potent, yes. But the reality is, if they're already broken, you know, sometimes just making a bigger hammer doesn't work. You need to change from a hammer to a socket driving wrench or find some other way. And that's kind of my simplistic way of looking at it. So we're actually launching an internal initiative to look at that using young blood and young plasma components, because there's now work showing that there's something in the plasma of young individuals that apparently can reverse the senescent and age-related markers found in cells in vitro. And there's a clinical trial going on in this now, if you can believe that. A company got a ton of money for it. I'm being waved at, so I better get out. We're also a single site. As was mentioned earlier, we have CGMP compliance. So the reagent products we can give you are going to be, you know, robust in terms of their tracking, their documentation, and the quality and reproducibility of them that we generate for you to use as a researcher. So y'all come see us. You know, we're happy to help out. We're a nonprofit. Money doesn't go into a hedge fund manager's pocket. It goes back into the blood bank and back into the community. So we're a pretty decent group to work with, and we would love to see folks come and knock on our door and work on this. Not only our blood bank, but blood banks in general, you know, in the U.S. I think it's an underutilized resource, cord blood banks and blood banks, and getting the word out to the research community that we're alive, we're well, and we're ready to help, I think is going to be critical to sustainability for blood banks, because we need to do something other than simply make blood and give it out. We're going to go out of business if we just do that. I'll close with that, and if there are any questions afterwards with the panel, y'all can come up and yell at me. Thanks.
Back on. Thank you, Dr. Tressler. Our next speaker anchoring this session is Charles Mooney, Vice President of Quality Management and Biodevelopment at Oklahoma Blood Institute, where he has been contributing to that organization since 1996. He fills many roles there. He's been instrumental in designing some of the very creative ways that they have grown their organization and started to interact with the research community, everything from licensure to creation of new business options, new business lines. So I'm going to ask you to welcome Charles. We'll get his presentation up. And then we will have the entire panel up here to address any questions you have.
Okay, so So I hope that by now you're beginning to hear the message that blood banks are probably a little bit more sophisticated organizations than what you might sometimes think. We're often known for our cookies, T-shirts, and community blood drives, but there's a lot of science going on at your community blood bank as well. You know, our day job is that we collect whole blood, we collect red cells and plasma and platelets, and we give those to millions of people throughout the country every year. We've been doing that for 50, 60 years here. But another thing that many blood centers are doing mononuclear cell collections. Is that me that's giving that feedback there? Do you guys hear it, or did I just hear it? Okay. Okay. How far back? One of the things that I was mentioning is that many of the centers collect mononuclear cells. We collect those by apheresis. We collect those through cord blood, and those are used a lot for bone marrow transplantation, but a lot of them are used for research use and are used for raw material for other cell therapy drugs. And we're going the wrong way. One of the things that you may not realize is that we can take those mononuclear cells that we collect, and many of us have technology like the BioSafe cell separator, and while we have those in our facilities primarily to do depletion of cord blood and volume reduction of cord blood, there are many other protocols that can be added to those devices, including, whether you know it or not, a FICOL separation. So there are a lot of capacity within blood centers and to do separation techniques. I don't know why my thing is on the control. Up is down, actually. Okay. Apologize for that. Let's start back to here.
That's my fault. Computers hate me. Delayed response.
Okay. Let's don't use the thing there. Okay. So we'll just use the up and down arrow instead of the upside down clicker that goes with it. So the thing that I was trying to communicate to you is that other than just the cell separation technology, some of us have the capacity to do selections. We've got a Clinimax system, so we can take those cells and do individual isolations there. We brought that into our facility because we had the need clinically to be able to do T-cell depletions for haplotype transplantations. Therefore, we've got a whole bunch of T cells that we're taking out that's now either can go in the garbage or can go into your research laboratory to be used for other things. We have the ability to add different types of columns to all of our apheresis technology. We have facilities that have hollow fiber on the quantum system bioreactors, so we can take a product, we can collect it, we can select it, we can isolate it, we can expand it, and of course in our blood centers we've been freezing, control rate freezing cells for decades here. So we have a lot of experience in freezing cells. We also have a pretty good bit of experience. Obviously, the cells that we are freezing, we're doing those for clinical use, so we want to be able to know that those are viable cells. And I don't know about in your facilities, but we don't do trypan blue in our facilities because we don't think that it's a good assay. So we brought in through our flow cytometer, the 7AD assay. We have the STEM Vision system there, which is a automated colony counting device, and many blood centers have those. We're used to doing flow cytometry for CD34 counts, and have recently come back and added, since we are isolating T cells now, then we need to be able to identify what we have. And so with those, that flow cytometer, we can now do CD3, 14, 19 testing. So that's available. Don't know if you are aware, but we've been culturing every platelet pheresis product the whole industry has for about 15 years. So we've got a lot of experience in doing cell culture or culture for bacteria, contamination culturing. We culture all of our cord bloods. We culture all of our peripheral blood-derived stem cell products, and so we're very comfortable doing anaerobic and aerobic cultures, and we have laminar flow class 100 hoods to be able to do that in. Also, since many of us are supporting transplant programs, then we have the ability to do high and low resolution HLA typing. So some blood centers just do HLA typing for class 1 antigens because they are finding compatible platelets for transfusion. Others are doing class 1 and class 2 and high resolution because they are supporting bone marrow transplant programs. As already been clearly mentioned here, but since I'm a quality guy, I can't get off of the stage without mentioning quality in some aspect here. And one of the things that I have as our center has begun to work with new groups is that there is somewhat of the misconception that good manufacturing practice is a clean room. And I've gone into a lot of facilities that have communicated to me that yes, we do GMP manufacturing, and they take me immediately to their clean room. And all the way on the way to the clean room, my eyes are just bugging out of the things that I'm seeing that are totally not in control there. So, you know, the thing that we have been doing for many years, because blood is a prescription drug and it is regulated by the Food and Drug Administration, we're very comfortable with the 200 series of the CFR because we've been doing hematopoietic stem cell collections for many years. We were the first adopters of the 1271 series of the CFR. We use a lot of medical devices, so we're pretty familiar with the 800 series there, and we are pretty comfortable with the IND and NDA process there. So blood centers do have a lot of regulatory experience, as has already been said. But if you don't hear anything else that I've said, period, one of the things that you need to step back and take a look at is that we have been doing cell therapy for about 75 years here. And so one of the things that we've been able to do through that 75 years of experience is, one, you may not realize that the blood center in your state is probably, unless you live in a high biotech region, in most states the blood center is the biggest biotech company that's in their state there. And most people don't stop and think of their blood center as being a biotech company that has a whole lot of infrastructure and a lot of experience in collecting, characterizing, storing, and distributing cells to people. And so, you know, one of the things that we have seen as we have gone in and looked at things as they are in the preclinical and then moving into clinical manufacturing is that there are a whole lot of steps that are involved in the manufacturing process that probably somehow have to come out of there to ever have a scalable manufacturing system that is going to allow you to make any profit for the science that you are developing here. And I will say another thing, that as I've been in this conference and looked at probably 150 new applications for cells that are out there, one of the things that you need to be thinking about is, okay, if I think that I'm going to get this beyond allogeneic, then how am I going to source those donors? You've got 150 different drug companies that are being developed that are going to be competing competing for the same space of donors there. So you probably want to begin to think about that right from the beginning here, and how can I use a portion of a community service that already has the infrastructure to do that, instead of me going out and trying to build a system that I would compete for those donors. So one of the things that you have heard said are the qualifiers of some blood centers, many blood centers. A blood center has the capacity to do something. So I put all the blood centers. I hope you can jot yours down. It's about a 8-point font here. This is all of the blood centers by state. There are 192 of them listed there if you just want to count those. So you might ask the question, well, how in the heck am I supposed to find which one can do what I need them to do? So glad you asked that question, because just happen to have a solution for you. Is one of the products that is going to be launched in 2016 is a new website called BioSharing. And what we are doing is going to the blood centers that have the capacity to be able to source these type of materials and to perform these type of assays and develop these type of assays, and to really be able to partner with you as an organization. And we're trying to pull all of those core competencies together into one space, so that you don't have to try to search down, this blood center is really good at HLA typing, this blood center is really good at cell collecting, and this one I need to be able to engage to understand what type of quality system that I need to have. The thing that we are trying to do is to pull all of those together into a standard location where they all can be offered through one website, and you can go there and customize what you need. So just for an example here, chose red cell because none of you probably are using those. So you would come to our website, you would see a description of what we have available, and everything is going to be customized. One of the things that you will find out as you go to the FDA and begin to have conversations with them is that they want primary source cells to be used. And so we really encourage you to utilize your community blood bank as a primary source cell provider instead of potentially using a cell line that that has problems that other people have already found and the FDA is very aware of as well. So with this site, you would go through and you would mark the characteristics that you want. You would place that request, and then we are going to decide which scientist within our groups, which center within our groups can best understand and meet this need here. And then we're going to call you. We're going to make sure that we understand, what do you need, what are you trying to do, you've requested this, but is this really what you want in the end here? You know, one of the things that we go back and look at some of the companies that have not made it, and as soon as they came to our blood center, we were able to tell them, wish we had had this conversation earlier, because you're starting with a raw material that's got too much variability with it. Had we talked earlier, we could have taken some of this variability out for you. And so that's one of the things that we are going to commit to through this site here, is that if we develop a protocol for you, then if we have to take that all the way across the country, that it's going to be a standardized protocol for you. So that variability can be taken out. So that site coming soon. One of the things that you may have noticed on that site there was right in the center is human subjects, and along with that site, we are launching another site called BioLink. And BioLink is going to be somewhat a combination between a dating service and LinkedIn. So when donors come into our facility and donate, and not just our facility, but every facility that partners with us, so we're looking to add blood centers across the country and build a massive database of information about these donors. We're not going to collect any samples. We're not going to to have any inventory of information or of a product. All we're going to have is bioinformation about these people. So the thing that we're doing that's a little bit different than anybody else in the market is doing is you come in, you donate, we email you a link to our website here. We ask you the question, you're interested in doing research, yes or no? You say yes, you get a link to LinkedIn. You can create your own bio profile there. You create it and you manage it. And so one of the things that helps with is that you don't have the consenting issue of whatever the IRB is going to require for your specific research. So what we have done is that we are going through all of the databases in each of the blood centers. We will search those for the characteristic that you're looking for, and we will say, yes, we found 25 of those for you. We will contact those people and find out, are you interested in this company contacting you to participate in this research? So if they are willing, then we will supply the connection there. Then you consent using your IRB approved research for the research that you're going to be doing. The beauty of that is that it's not de-identified, but it becomes under your control in your research, and we can also go back to those people over and over. And so that's one of the things that we think that we're bringing to the market that currently doesn't exist. One is a broad spectrum all the way across the country of potential research subjects that we have collected the same information about, and the ability to go back to people and potentially be able to get B cells and T cells and dendritic cells all from the same cell source there. So look for these two things in 2017. BioLink is actually out there if you know how to get to it, but BioSharing is not yet. So if you want to go out there, bio-linked.org, and you can go to that website. Okay, thanks.
Please speak to the necessity or just your thoughts about quality systems and the ability or the necessity of I guess getting those together.
Yeah, great question there. I think that my response would really be twofold there. One is that you need to know that whoever you're getting your source material from really does have a true quality system in place, because that is going to be audited. When the FDA starts to license your drug, then they are going to come and inspect us as well. So you need to make sure that whoever you're partnered with, that you know what's in their closet. The other thing is that there needs to be real consistency there. Is that one of the things that I have heard over and over from researchers and developers that are looking at mononuclear cells specifically collected by apheresis there is the wide diversity in the product and how it was collected, and the FDA is not going to be very forgiving of that. That process needs to be standardized, and everybody needs to be. If somebody's collecting cells for you, they need to use the exact same protocol to collect those. And then the other thing that I would say is we really wish that you would use some of the pain that we have gone through over the last 30 years and putting these quality systems in place, to use that experience to let us help you develop some of the procedures and some of the validations and some of those verifications that we know that the FDA is going to look for when they inspect your facility.
Linda, any additions from your quality background?
I would say that we are looking at collaborating with other blood centers to try to standardize some of our quality systems. We all have some pretty robust quality systems in place, but I think moving forward, being able to standardize across multiple advanced blood centers would be a very desirable thing to do, and we have some initiatives in process along that way.
I'm wondering how the work in cell therapy and regenerative medicine has changed how you're thinking about your customers. Are you segmenting them differently? Are you going after new customers or seeing a need to serve new customers? How is that working in your organization?
I think one of the things there that we have to be somewhat proactive at doing as blood banks is realizing, you could say, well, I'll give a specific example. How much of a market is there right now of people pounding on the door for expanded CD34 cells from a cord blood unit? Well, there aren't a lot of people pounding on the door, but it's not because there isn't an interest and a need. It's because, frankly, the products are just now being developed. It's kind of the argument that was made hundreds of years ago. Well, I don't need an automated system for printing books because I can write. And so there wasn't a demand for printers until someone made one and everybody went, wow, okay, everybody wants a printing machine now. I think we need to, as blood banks, is to be creative and insightful and look at what the community is doing, look at what the research community and the drug development community is doing, and say, where do we fit in with the resources and the potential products we have, and then start making some forward momentum internally on that, and then presenting that to the community at large. Instead of speaking conceptually about, well, we could do this or we could do that, say, look, I've got, 10 billion CD34 cells that are HLA characterized, and I can give you low, intermediate, or high resolution for you to do research with, or you to do preclinical development or translational science with. Generate the products, position them, and make the community aware. And I think that if you build it, they will come. I think if we're passive and we kind of wait, first off, there's a lag time to develop the product because you do have quality, and quality keeps me out of the woods. Thank you very much. Quality and regulatory are integral parts of that, and that's one of the advantages that that blood banks have in general, is we already have that robust quality infrastructure. So when we generate products for you, they're going to be good products. They're going to be properly reviewed, consented if need be, if relevant, and IRB cleared if relevant. But I think the onus is not only on the community at large to try to develop therapies, but on the blood banks themselves to reset their mindset, to think of those products that we think would have utility and start working on generating them. And I'll get off the soapbox.
If I could add just one thing to that is, as we look across the systems and the drugs and the devices that are being developed, it's a little overwhelming because one of the things that we have a responsibility to is our hospitals and our communities there to make sure that our hospitals and our communities have access to these drugs in the future. And so, if you're an individual laboratory and you're working on an individual drug there, then you have the luxury to be able just to focus on that. And you really don't have to think about, well, what happens if there are 18 drugs that use mesenchymal stem cells? How can all of those be available within my community and be available to all communities throughout the U.S.? And so, I think that one of the things that I would really encourage people as they begin to think about right from the beginning is how am I going to be able to compete in this space long term, and how will I be able to manufacture this in a way where whatever the reimbursement that insurance companies provide for this therapy is going to provide a margin that I can build a company around.
Okay. Other questions? What have you changed about your organization to accommodate this new area of exploration in business, whether it's infrastructure or hiring, skill sets?
We have done a lot of change of infrastructure and focusing and diversifying. I think that's key for blood centers and the blood industry is in diversification, and really segmenting and focusing on and putting resources to developing what we think is going to be the future. I really feel like we're pioneers at this point, moving into the future of a new type of medicine, and we're making investments into that, and most of us are not-for-profits, so that is a big challenge in finding funding to help us lay that that foundation so that we can make that leap into the future with where regenerative medicine is headed.
Sorry, and I don't want to pass up the opportunity to speak, so the mic was closed. The one thing that I would add there too, and it really comes back to this concept of being able to create manufacturing processes that are long-term sustainable. There is one of the things that we are trying to understand there is how do we put infrastructure in place that can be supportive in that without every facility that would make a mesenchymal stem cell drug or a CAR T-cell drug, why should every one of those put those same systems in place when there can be a community resource that could collect and isolate and then provide you a better source raw material. So I think that the thing that I would encourage again people to think about is start with the end in mind and say, okay, well, how much of my operation can I push back to that community blood bank as closer to the patient there and transfer those costs to a variable cost as I need them instead of me having to go and raise a lot of capital to build an infrastructure that really I could just rent from my community blood center.
So as you've made some of those changes, I'm wondering what has been the most challenging obstacle to getting those in place, and if you're looking forward to the next 3 to 5 years or even 1 to 3 years, what do you see as the biggest hurdle you're going to need to clear?
So my mother said, you were given 2 ears and 1 mouth. I wish you'd learn to use them in proportion, but hasn't worked yet. Internal education, at least for my experience, has been, as part of it is, blood banks are wonderful organizations. I've been donating blood since I was a little kid, pretty much. By that meaning under the age of 29. A lot of it for us initially was there was not resistance, but there was kind of an inertia to building out a research facility, an actual non-GMP basic research facility. We actually have that. We go everything from non-GMP to full CGMP ISO qualification capabilities. So depending on the researcher, we can give you a hybrid of just pure basic research opportunity and access all the way out to you're going into the clinic and we're here to be audited and be an advocate for your product. But internally, there was a lot of, well, we've done it this way for decades and it's worked well. If it ain't broke, don't fix it. Well, the blood banks aren't broke, but the environment we work in, economically and medically in terms of practice, has changed. Okay? The weather is changing. It's getting colder or hotter depending on your perspective. And if you don't adjust accordingly, you're going to have a tough time of it as a blood bank. So part of it was an internal education. The second part of it, now that we've gotten that out of the way and people have bought into it, robustly so, I'm glad to say, and I'm thrilled to see this kind of a venue. This is a reflection, I think, of that, is now not just talking about this at the World Stem Cell Summit, which is a great place to start, but now getting out into the larger research community and stem cell therapeutic and biopharma community and letting them know about us as well. I think that's the other hurdle that we're starting to clear that we really need to get after, though.
Yeah, I agree with the internal. I know at our facility, the whole concept that you can do research and it is different and the regulations are different and you need to sort of have a separate group or area to do that was a hurdle that we kind of faced early on and we've been developing for some years. And I agree with what was just said, that that's very important in order to have that. Like we already mentioned, we have a very strong quality background, so sometimes it's kind of hard with quality to say, hey, I'm going to do something a little different. So there are actually different quality rules I need to follow over here, and these other ones don't pertain. And I know it takes a little bit to get that knowledge across and sort of that understanding that there are different areas and different things you have to do with that. And I would say the other thing that was already sort of mentioned is really going out there and really going face-to-face and meeting with facilities and making the time to go out there and really go door to door and talk to people and say, what are your pain points? What can we do for you? And like was said a minute ago, I agree with the whole concept that if you go out there and say, hey, here's what I can do, yeah, they'll write it down, but you're not going to get anywhere. If you don't go out and listen and then say, hey, here's possibilities, or hey, you've got this great product, this would work great with our tissue center, or this would work great for micro and save us and enable us to have a competitive advantage. Those are the type of things that you really have to do in order to make that system work. So.
Actually, I know this isn't relevant to the question per se, but that's one of the things we have to do to the customers, the research customers. We have to demonstrate that we're going to be an advantage for them, whether it be by assay development, source material, quality, or even cutting-edge science. I mean, there's nothing illegal about doing good science beyond what we've classically done there.
Great, thank you. So I've given you in the audience some time to think about it, but I'll pose the question again to both the panel and the audience. As we've talked about these different ways to collaborate, different ways to think about the resource that a blood center offers, blood and tissue center offers, are there any ideas or needs for collaboration that you're experiencing right now? Just in case there's someone here who can address that need. All right, the crickets win.
We're open to all collaborations, as I'm sure our colleagues all are here. We have some esteemed people in the audience, and if there are some comments, we'd love to hear them. I think we would all be interested in it. I think we've got some wonderful opportunities now and moving forward, and developing and partnering, and I see great things in the future.
Great. Thank you, Linda. Thank you to the entire panel. And thank you for attending and sticking it out till almost 2 o'clock on Saturday afternoon. With that, we'll adjourn for the day.